My name is Yasmin Kusa, and today I'll be presenting my Journal Club article selection. My chosen article discusses the ways in which various non-nutritive sweeteners can induce structural and functional changes in gut and oral microbiomes and lead to changes in glucose tolerance. This study aims to answer two main questions at the same time. Firstly, can non-nutritive sweeteners, also known as artificial sweeteners, affect human glucose tolerance? And if so, does the human microbiome impact this in a person-to-person manner? It is important that this question is answered as artificial sweeteners are widely gaining popularity as healthier alternatives for more caloric, naturally derived sweeteners and are often used in the management of issues such as diabetes and obesity. However, in order to truly determine if they are a better option on overall health, their effects on blood sugar must be studied to understand the safety and efficacy of them. More importantly, we must also attempt to understand the extent to which an individual's microbiome can impact the way in which sweeteners affect blood sugar or glucose levels. To begin, it is important that we define some key terms and ideas. Firstly, the term non-nutritive sweeteners refers to the artificial sweeteners that are marketed as being 0 calories and are popular alternatives as they offer little to no nutritional value, such as added sugar on food labels. Examples of non-nutritive sweeteners include acesulfame K, advantame, aspartame, neotame, saccharin, and sucralose. An article published on the American Heart Association's website discusses these non-nutritive sweeteners and recommends consuming them as an alternative to food and drinks with added sugars in an effort to lower blood glucose levels, manage diabetes and weight, and lower the risk of becoming obese or developing heart disease. We can see these artificial sweeteners in things like diet sodas, sugar-free energy drinks, sugar-free desserts, low-calorie syrups, some protein bars, and protein powders, just to name a few examples. We can truly see these non-nutritive sweeteners implemented in plenty of food products on shelves nowadays. In this study, researchers focused on 4 non-nutritive sweeteners: saccharin, sucralose, aspartame, and stevia. However, research such as this study suggests that non-nutritive sweeteners have the capacity to potentially disrupt the gut microbiome, which could in turn impact glucose metabolism further. In 2025, the FDA doubled down on an external safety review of aspartame that had found it to be carcinogenic, in which the FDA reiterated the safety of consuming artificial sweeteners at the current acceptable daily intake quantity. However, a secondary study found that non-nutritive sweeteners exemplified some capacity to alter the microbiome composition and metabolic pathways in the human duodenal lumen, which is a portion of the small intestine that is key to nutrient absorption during digestion. With these key points in mind, it is important to call into question the safety, benefits, and disadvantages of relying on non-nutritive sweeteners as a dietary alternative. Although deemed safe and are popularly used in a variety of food and drinks, our understanding on their impacts on the microbiome is very much limited. While there have been a few other studies that have attempted to answer these same questions, this study really tries to go the distance in comparing multiple non-nutritive sweeteners simultaneously, while measuring their effects on microbiomes and blood glucose levels in human participants and mice. Let's discuss some of the specific aims that this research attempted to target. First and foremost, researchers wanted to determine if these non-nutritive sweeteners had the ability to impact human metabolism. Specifically, researchers set out to measure glucose tolerance, which is a measure of how well the body can process glucose levels in the blood over time. This test will be able to tell if non-nutritive sweeteners impair glucose metabolism in any capacity. Secondly, researchers also sought to determine if these sweeteners altered the composition of the human gut microbiome. Through their series of experiments, this could reveal if any structural or functional changes were present within the gut microbiome following consuming these sweeteners after an extended period of time. Doing this would test to see if there's a direct correlation between microbiome changes and metabolic outcomes in humans. Third, if researchers were able to establish a causal link between non-nutritive sweetener consumption and changes to the gut microbiome, then they wanted to take it a step further and see if these specific changes were personalized to an individual's microbiome. This means that they wanted to see if a person's individualized microbiome structure prior to the experiment impacted these responses. And finally, the experiment was designed to test multiple different non-nutritive sweeteners in parallel to determine if each sweetener produced a distinct outcome or if they produced a similar effect to one another overall. All in all, this was a pretty large experiment that sought out to answer multiple questions. However, given how interconnected microbiome structure and metabolic pathways are, a complex experimental design was the best way to set out to answer these many important questions. Now we will discuss the specific methodology used in the study. The experiment can be broken down into two main phases, the first being the controlled human experiment and the second being the experiment involving germ-free mice. Starting with the controlled human experiment, 120 healthy adult participants were initially screened and selected to be involved in the study. The individuals all selected were in good metabolic health, had a normal BMI, waist-to-hip ratio, hemoglobin A1C, cholesterol, blood pressure, heart rate, serum alanine transaminase, and aspartate transaminase. So pretty much all the participants were deemed very healthy to establish a good foundation for data collection. The participants were separated into 6 groups consisting of 20 people in each group. Each of the groups were assigned a specific non-nutritive sweetener. One group was assigned stevia, the second was aspartame, the third was sucralose, and the fourth was saccharin. The fifth and the sixth groups served as 2 control groups. The fifth group was assigned powderized glucose vehicles to serve as the caloric control group, and the sixth group was assigned no sweetener supplement at all. The data collection in the human portion of the experiment took place over the span of 4 weeks. On day 8 through day 21, the participants were instructed to consume 6 total packets of their assigned sweetener daily. Each packet contained slightly less than the acceptable daily intake recommendation for each sweetener type. Participants had to dissolve 2 packets in water and drink it 3 times daily in the morning, afternoon, and night, and it could be taken with or without food. Now let's talk about the ways in which data was collected from human participants. Firstly, every single day, each participant had to log all of their food, liquid, and instructed sweetener intake on a digital platform on their smartphone. These logs were checked every single day to make sure everyone was adhering to the study's strict protocols. Additionally, the study wanted to look at some very specific biomarkers and microbial compositions of the gut and oral microbiomes in each person. Here are some of the ways in which they were able to achieve that. Their oral microbiomes were able to be profiled by instructing each person to swab their buccal cavity, also known as their inner cheek, in the morning after having brushed their teeth, but before eating anything or using mouthwash, and this only occurred on certain days of the trial. To survey their gut microbiomes, stool samples were also collected on certain days of the trial as well. So in addition to logging their food intake daily, they also had to record all of their daily physical activities and/or workouts on the same log. The study subjects had to also be connected to a continuous glucose monitor for the duration of the experiment in order to have access to their blood sugar levels at all times of the day. Additionally, on day 0, day 14, and day 28 of the experiments, each person had their blood pressure, heart rate, anthropometrics, and blood samples collected by a research associate or a nurse. ELISA testing was also used to look at each person's insulin and GLP-1 levels. And lastly, at-home glucose tolerance tests were taken at 9 instances during the trial period, while the participants were in a fast state to further test the metabolic outcomes of non-nutritive sweetener consumption. Now, let's quickly pivot and talk about the portion of the experiment that involved germ-free mice. Why were germ-free mice even used in this experiment? Well, germ-free mice serve as an important study model since they lack a distinct gut microbiome and are raised in a sterile environment. When human stool samples were collected during the human portion of the experiment, the fecal samples were transplanted into the germ-free mice to transfer over the microbiota found in humans. After this transplantation occurred, the mice were fed a regular diet and were monitored for 1. Blood glucose changes, 2. Microbiome composition, and 3. Metabolic changes. This was done by using some of the same testing measures as they did for humans, such as also performing glucose tolerance testing, collecting fecal samples from the mice and analyzing them, and monitoring their biomarkers as well. After having collected all of the oral and gut microbiome samples from both the humans and mice, Sequencing techniques were utilized to draw an accurate picture of how microbiome structure is impacted throughout this trial. This paper utilizes Illumina NextSeq technology for metagenome shotgun sequencing in order to analyze the genetic material found in oral and stool samples. We learned that the Illumina platform is best for producing a large abundance of short DNA reads and high-quality bases while being cost-effective. Another benefit of using Illumina is that it has a very high throughput, meaning it produces a large amount of data that can be analyzed to look for very subtle changes in microbial structure and the genes present across the test groups. While this is not technically a sequencing method, another form of analysis was performed called metabolomics. This method is used to analyze metabolites extracted from the collected blood samples, such as lipids and amino acids. Mass spectrometry was also used to achieve this. The purpose of this additional analysis was to figure out what metabolite products were produced and how they may induce changes to glucose metabolism further. All that being said, now we can finally move on and talk about the results. I would say there are actually some really interesting key findings here. Let's start with our first key finding. The experiment revealed the human test groups consuming saccharin and sucralose experienced a detrimental effect to their glycemic responses over time. This figure summarizes the overall results of the at-home glucose tolerance tests that all of the human participants were instructed to complete during different points of the experiment. Figure 2a of this figure shows the p-values obtained when comparing the test groups with sweeteners versus the two control groups. We see that saccharin and sucralose are the only groups that both display p-values less than 0.05 when compared to both control groups. In statistical analysis, p-values less than 0.05 highlight statistical significance. This already begins to single out these two non-nutritive sweeteners as influencing glucose significantly in a negative way. Figures 2b to 2d use the following graphs to measure the IAUC value, which stands for the incremental area under the glucose curve, and is often a marker used to assess the body's response to glucose after eating. Each of these three graphs plot the average IAUC value for each participant in each group across 3 periods of time during the trial, where each dot represents one individual. These interesting-looking, colorful shapes are supposed to convey changes in the distribution of data over time, and again we can see the largest fluctuation in shape in the groups assigned to sucralose and saccharin. And lastly, figures 2e through 2j give a closer look at how the average IAUC values for each specific group compare at different points in time. While aspartame, stevia, and the two control groups only showed a slight linear trend, sucralose and saccharin show big jumps in glycemic response that return closer to baseline after the trial ended. So to wrap up with this figure, 2 major things we learned from this alone is that 1, non-nutritive sweeteners do in fact have the ability to impair glucose metabolism, and 2, not all non-nutritive sweeteners result in the same metabolic outcomes. Instead, they have distinct effects, with sucralose and saccharin being the most detrimental. Okay, now that we have established the correlation between non-nutritive sweeteners and glycemic responses, we can now try to explore a link to functional microbiome alterations. For this portion of the results, we will discuss the findings discovered in both the human gut and oral microbiomes. Let's start with this figure that deals with the human gut microbiome first. Just as a reminder, this experiment collected longitudinal stool samples and conducted shotgun metagenomics to be able to profile the microbiome composition of the intestine. So based on the data, the baseline stool samples collected in the beginning of the trial were all pretty consistent in structure with one another. However, over the coming weeks, changes in the microbiome structure for the 4 sweetener groups were observable and was most significant in the sucralose group. These 6 graphs show a trajectory analysis of some of the non-nutritive sweeteners and are supposed to highlight if there are any significant changes found, and we can see that in figure 3a that sucralose presented the largest shifts in the gut microbiome. In fact, all 4 non-nutritive sweetener groups displayed some alteration in microbiome structure, with some groups having a greater effect than others. When the control groups had their microbiome compositions tested via stool sample, no significant changes in bacterial genera was found. Let's continue this discussion by now reviewing the observable changes in the oral microbiomes tested. Once again, the two control groups showed no significant difference in oral microbial composition. However, each of the 4 non-nutritive sweeteners caused a different bacterial strain's abundance to fluctuate or induce metabolic pathway changes. Figures 3a to 3d show how stevia impacted 4 metabolic pathways by decreasing their rates in the second week of the trial. Figure 3e shows how the sucralose group experienced a variety of changes in the relative abundance of Streptococcus species. Figure 3f shows a reduced relative abundance of Fusobacterium in the saccharin group. Lastly, Figure 3g shows a reduction in both the abundance of Porphyromonas and Prevotella nansiensis strains in the aspartame group. These last two figures are highly significant indicators of the impacts that these non-nutritive sweeteners can have on both the gut and oral microbiomes in humans. Not only were there structural changes found, but the functional changes were present as well. This is a huge telltale sign that microbiome alterations also induce disturbances in multiple typical metabolic pathways, which was also proven in the changes to glycemic responses during the trial. Furthermore, this figure shows a link between metabolome changes and altered microbial activity. Each non-nutritive sweetener showcases a distinct impact on metabolic pathways. We see a large shift in the manner of gene expression between each group. The left side shows how each group experienced changes in metabolic pathways and bacterial species abundance. To the right of each graph, we can see how plasma metabolites increased as well for each group. This figure should be analyzed row by row to understand it a little better. This figure can be really overwhelming to look at and reveals so much information about many different changes that occurred metabolically and in microbiome composition. I will attempt to summarize everything to the best of my ability. So here are the major findings. In the sucralose group, 22 plasma metabolites including short-chain fatty acids, propionate, butyrate, and valerate were determined to have a direct effect on metabolic pathways like the TCA cycle and amino acid biosynthesis and metabolism. All of this in turn showed a significant link to impaired glycemic responses. Additionally, researchers also wanted to prove if and how changes in metabolic pathways may differ from person to person. They compared the 5 individuals in the sucralose group that experienced the biggest impact during the trial versus the 5 individuals that experienced little to no change. The key difference was the presence of 3 metabolites: beta-hydroxybutyrate, serine, and cysteate. The 5 individuals who had the most severe glycemic response were lacking in these 3 metabolites. This goes to show that additionally, individual variations in metabolites and microbiome composition can induce varied responses. Similar personal responses were also observable in the 3 other sweetener groups. The saccharin group experienced bacterial changes and was linked to increased indoxyl sulfate and decreased long-chain fatty acids. Stevia shifted levels of arginine metabolites, ornithine and citrulline, in addition to other amino acids. And lastly, the aspartame group increased kynurenine. Once again, both control groups displayed no changes such as these. I know that was pretty extensive, but it goes to show just how much of an impact these non-nutritive sweeteners have on dynamically altering the human microbiome, and thus metabolic pathways on a very personal level. This last figure I will be presenting once again reiterates the same overarching concept as the last figure did. However, this figure presents the individual outcomes found in germ-free mice. This data was produced by taking stool samples from 42 humans, consisting of the top and lowest responding participants to the treatments. The stool was transplanted into mice. Remember, the mice were fed a regular diet and not given the same sweetener supplementation as humans. However, what is really interesting here is that the same glycemic response changes that occurred in humans also occurred in the germ-free mice, without them even having to consume any sweetener. This means that the microbiome changes that were induced during the human trial had the ability to continuously influence mice by causing glucose intolerance in them too. The mice that had the feces transplanted from individuals in the control groups showed no change in glycemic response. This reinforces that not all non-nutritive sweeteners act identically. Additionally, The mice portion of the experiment supports that individual differences in the gut microbiota can influence how we can respond to non-nutritive sweeteners. Lastly, this proves that the microbiome is a key mediator in the glycemic effects that non-nutritive sweeteners can have. So now that we have finished discussing the results of the experiment, let's circle back to the initial questions I brought up in the beginning of my presentation. Can non-nutritive sweeteners affect human glucose tolerance? And if so, does the human microbiome impact this in a person-to-person manner? These questions lead us to key conclusion number 1. Non-nutritive sweeteners have personalized and microbiome-dependent effects that can induce glucose intolerance. With all of the presented data in mind, we can confidently determine that non-nutritive sweeteners do in fact induce changes to human glucose tolerance. We saw that all 4 groups were able to induce some level of change, with sucralose and saccharin inducing a detrimental effect on glucose tolerance. However, there was variation in the amount of change shown from person to person due to their individualistic gut microbiomes. 2. The gut microbiome alone can also play a causal role. We were able to find this out through the experiment with the germ-free mice, where they showcased impaired glucose tolerance through simply being transplanted with microbiota from the human participants in the experiment. This reveals that changes to the gut microbiome alone can be enough to alter metabolic regulation in both humans and mice. Number 3. Of the 4 types of non-nutritive sweeteners, sucralose and saccharin impose the greatest number of detrimental effects. We were able to see that sucralose impacted gene expression the most and altered key metabolic pathways, leading to the greatest disruption in glucose metabolism. It also further supports the relationship between the functions of the microbiome and metabolism. Number 4. Not every non-nutritive sweetener is the same in its overall impacts on a host. We were able to observe a great variation in the effects that each sweetener group induced on humans and mice. Each sweetener led to distinct alterations in microbiome composition and metabolic functions. Number 5. We cannot confidently deem non-nutritive sweeteners as being safe from a public health standpoint. The much larger discussion at hand that I described in the background section of my presentation discussed the uses of non-nutritive sweeteners as being an allegedly healthier alternative to more natural caloric sweeteners. I find it somewhat concerning that organizations like the FDA and the American Heart Association tell consumers that they are safe for consumption and are a reliable method to manage numerous health problems, most notably obesity and diabetes. Studies such as this one challenge these organizations' public health recommendations and bring light to a much larger debate about whether or not these low-calorie alternatives are truly healthier for us in the long run. To continue this discussion, I would like to talk about some strengths and weaknesses that this study may have, as there's always more questions to be answered. Some of the strengths including 4 types of non-nutritive sweeteners, open the door for a comparative analysis. Number 2. The uses of metagenomics and metabolomics revealed a multitude of data on the various changes induced. 3. Using germ-free mice as a secondary test group validated causality further. Number 4. The strict protocol ensured accuracy and reliability within the set of collected data. Some of the weaknesses include, number 1, the test duration was very short and limits the understanding of long-term non-nutritive sweetener consumption. Number 2, the sample size was relatively small. Number 3, the sample only included healthy adults, so the study was not very representative of the overall population. And number 4, there are other types of non-nutritive sweeteners outside of the 4 tested, so we do not understand their impacts on health. This study was able to prove that non-nutritive sweeteners do in fact induce glucose intolerance, namely saccharin and sucralose. Additionally, we have determined this is done in a microbiome-dependent and highly individualized manner. Since the gut microbiome plays a significant role in mediating these effects, we cannot say for certain that non-nutritive sweeteners are beneficial for all people. Additionally, this study draws attention to potential areas of concern within the use of these popular sweetener alternatives. We must continue to take a closer look at their effects on holistic health. We must also advocate for better nutritional guidance given the variability of microbiomes between different individuals. This study was a great stepping stone at exploring this topic. Going forward, it would be helpful to see some more studies done involving larger, more representative populations for longer periods of time. Additionally, finding ways to mitigate the potential harm done by these non-nutritive sweeteners would be significant in following up on this research. With the development of research on this topic, it also becomes important to note that this information may not stop people from consuming these sweeteners, especially if they have already grown a reliance on them within their diets and lifestyles. I know that for myself personally, I have relied on artificial sweeteners and consume them daily, so it is definitely difficult to implement wide-scale changes with all these new findings coming to light. I think greater transparency from large organizations would be a great start in educating the public about the potential negative consequences of consuming these sweeteners. Personally, I learned a lot about this out of my own personal interest in this topic, given that I am an avid Diet Coke drinker. So finally, here are a few questions that offer further discussion in regards to the study. Number 1. How do other non-nutritive sweeteners, not including the ones already discussed in this article, impact the human microbiome and its metabolic pathways? Number 2. If the study tested humans with pre-existing metabolic disorders, how could this have influenced the resulting structural and functional alterations of the microbiome? Number 3. What does this study suggest about the adaptability and resilience of the microbiome? Number 4. Could the supplementation of probiotics or prebiotics lessen the potential negative effects of consuming non-nutritive sweeteners? And number 5. How should the study influence public health recommendations around non-nutritive sweeteners, especially regarding diabetic individuals? Here are all of my references, and that concludes my presentation. Thank you again for watching.